scale on your graph?
I deliberately didn't put any, but it's 3, 4, 5 years.
Saving is 3, 4, 5?
No, the saving is about half. So saving is in years, at least. And it's also a saving that is substantial when you actually do the manufacturing. So you reduce the time both for building the facility and also when you do the actual manufacturing. So one more technology that I want to show you, and this is also connects to the vaccine business. So today, when you measure how much of the influenza vaccine that you have produced, you use a very old-fashioned technology from the 70s. It's actually an agarose gel. You have your antibody in that, and you punch holes in that gel. You add your sample into those small wells, and you measure sort of how big this ring around the punched hole is. So this is still the assay that is required by all health organizations to measure how much of your vaccine you have. And I think you can imagine, even though you may not be a scientist originally, the robustness and the precision and the accuracy of this assay is not up to today's standards. So we've used one of our protein analytic technologies called an equipment called BioCore, when you can actually measure the binding between these two individuals. We've shown that we can sort of increase the sensitivity about 10-fold, which means that you can actually measure the low amount that you have in a dose. Today, the old-fashioned technique only measures to a very high level, and the actual dose required is smaller. But you can't use that low dose because the assay don't match it. So using this technology, it's 10-fold more sensitive, and you can use everything you produce, so you can produce more batches. It can be used for treating more patients. And it's also significantly less manual requirements input to this. So it's sort of a standard up to date technology. It's automated and a high throughput of the technology. So it's one example of how the technology today can speed up development of new drugs, new vaccines. For this particular example, it's been used for influenza vaccines, malaria, and a couple of other so far. Speeds up development significantly. It makes that you can produce more doses, so it increases the access to a broader population. And the precision of the assay is so much better, so it increases the quality of the drug. So I think this is a truly good example of what we think the health imagination perspective, that challenge, how we respond to that challenge. So this was a few examples of our technology and the imaginative science that we can provide. I just want to connect, when we heard through all these speakers, that what we see, okay, we're a global company. Products are distributed over 200 countries. I have R&D groups in many countries. So why are we still present in Uppsala? We've touched upon it a bit here. I think we have, as I see it, a very good competence in the area. We've been in the life science space for many, many years. So there's a true competence in the area, and I can then have good expertise and people that I can employ. We also have a strong tradition of collaborating. And I think especially if I compare to other countries globally, in Sweden we can collaborate between the academic research groups and the industry. And it's put forward as a good thing to have this collaboration. And the spin out of small companies, it's seen as a positive thing to be able to translate academic research to a global use, to increase health, to commercialize these products and make sure we transform academic research to a commercial setting. So I think that sort of two of the two good examples of why we still are in Uppsala. I could have picked sort of any different region, but I'm pretty sure we will continue to have our R&D focus in Uppsala and continue to grow that facility and expand all the products we can provide. So I guess that opens the floor for any discussions, questions?
Do you have any plans to, in your field you're making tools for research, which is what you're doing. I mean, do you have any, why haven't you done what they've done? I suppose.
We're doing parallel things at the moment, and of course we're talking to Christian and his company, as long as many other companies, in how we can contribute to sort of transforming these technologies to a more commercial setting. I think the true machinery that we have is to identify technologies that are invented anywhere globally and transform them to commercially products that we can then distribute to a global setting. That machinery is something that we can provide. And we work with companies like Biolamina and others to be able to sustainably deliver these products.
So what proportion of the kind of innovations that you've introduced over the last, let's choose a period of 10 years, have come from Sweden versus Europe versus global research feed? You know, so when you adopt techniques and commercialize techniques, is it all from your own labs, or is it?
The vast majority is from the Swedish region, I would say. And I think it's lately we've broadened that perspective and added on sort of a more extensive search of technologies in a global context. But the very, very vast majority of the products we provide today are from innovation in Sweden.
Is that because you have a narrow outlook?
Sorry?
Is that because you have a narrow outlook historically?
No, I think it's because we've been so successful with the technologies we already provide. And with the long tradition of I didn't go to the history at all. We have about 50, 60 years of history in the chromatography business. It all started in Uppsala. And it's been very successful, and that's the basis of the entire company, and we since then sort of broadened the perspective and added on technologies. I guess it's the last 10 years or so that that's been sort of expanded.
I guess my point was, what have you missed by not scooping in innovations from elsewhere?
Yeah, you never know. You never know. And it's things might have happened 20 years ago or 30 years ago. I think it's what we see, why I think the protein science is real right now. It's so much happening now. It's moved to, it used to be Europe and US. Everything was happening. We now see lots of new technology that rise in sort of India and China, Korea, other types of regions. So the life science space is much more global today than it was 10, 20 years ago. Okay.
Well, I think with that,
Yep.
I think I've been given the privilege of summarising everything. So I've got half an hour. No, it won't take that long. I think thank you very much for all four speakers. I think we've had some really interesting talks here. There are a couple of things that came out to me, really. The first is, and I use this word guardedly, but it seems as though we're getting to a point where there is some level of standardization in looking at proteins. When we started off, and when I was in my cold room, it was one protein at a time. And I think that what we're beginning to see is some technologies that allow some better reproducibility. I hate the word industrialization, which is bandied around a lot in research, because I think there's a lot of innovation in research. But we're sort of getting more towards there being processes. That means that not quite turning the handle, but certainly you can do things with something that's a reproducible fashion. I think if you look at the areas of therapeutic antibodies, without some of the work on protein A, which is one of the key things for purifying proteins, which comes from Sweden, from pharmacia, that whole field just wouldn't exist. Well, or maybe someone else would have invented it, but certainly it's facilitated what's going on in antibodies today. We didn't even talk about things like factor VIII, which is an important protein for haemophiliacs that, I don't know when they started making it in Sweden, 25 years ago?
It was a commercial drug in '99. It's been in development since the beginning of the 90s.
So there's a long history, I think, in Sweden of producing protein drugs and helping to purify protein drugs such that they can then go into patients. I think the other thing to remember, I was one of those people who got very excited about genes, well, many years ago, but sort of the sequencing of the genome 10 years ago. The key thing is proteins perform functions. And I think one of the things that I think Christian's talk, we're beginning to learn about a particular class of proteins that can perform a function that can help in stem cell research. And so from that, you come up with a tool that actually, it looks to me, is going to be essential for propagating particular types of stem cell populations that then you can actually go and use them in the clinic, and you can get through regulatory processes. And so I think our understanding of how proteins work is beginning to get better, and there are applications of that. I think we learned a little bit about sort of proteins reflect what has happened or what is happening in the body. That's very different to having a genetic test at birth telling you there's a 30% chance you might get diabetes at the age of 55. What do you do with that? Whereas I think diagnostic tests that tell you this patient is stage 6 prostate cancer or stage 7, there's a clinical sequelae to that. Do I do something or do we just wait and see? And I think that's quite important in terms of real-world applications. And I think coming back to the research level, proteins are drug targets. So drugs act on proteins. And I think some of the work that we discussed a little bit in the Q&A session, there was a lot of excitement about membrane proteins because there are so many of them are drug targets 15, 20 years ago. But very little of that has panned out because we didn't have standardized approaches to understand their structures, which means you can then go and design better proteins. And I think we've learned a little bit today that we're getting to that point too. So if we go back to where we started in hype or reality, I think there is a turning point that's coming here. And I think what's interesting about sort of what we've learned today, what's going on in Stockholm and Uppsala, is that you need a multidisciplinary approach and you need to collaborate. And I think having doctors and scientists and engineers and IT guys all on sort of one site and actually talking to each other is the way forward. I think everybody would probably agree with that. So that's what I took away from today. I thought it was really interesting, and thank you very much for everybody attending. I think we're going to have some lunch now, and of course, everyone's ready for more questions.